RNA molecules are very unique molecules. This means that RNA is going to have multiple roles inside of a cell. We do want to make sure that we remember the differences between RNA versus DNA. And if we just point out quickly some of those differences, RNA molecules have a different sugar, so there's a sugar difference. There is also going to be a base difference. So we have uracils in RNA rather than the thymine. And another thing to keep in mind is that RNAs are going to be single-stranded molecules. Now, because they are single-stranded, that's going to enable them to take on a lot of unique three-dimensional shapes. So that's really what we see referred to in point number 2 here. They take on those unique three-dimensional shapes because they can hydrogen bond to themselves. So the bases that are in the molecule, they're not already bound to something else the way that they are in DNA. So we get a lot of unique shapes, and we've seen some of those unique shapes already. Another thing that RNA can do is, again, because those bases are exposed, they can certainly hydrogen bond to other nucleic acid molecules. We do see this in several examples. Just to emphasize a few, we saw this during the splicing process. During splicing, we had the SN RNAs that recognized the junction between the introns and the exons. So they detected where an exon ended and an intron began. They did that by actually hydrogen bonding to the mRNA template molecule. Another place where we saw RNAs hydrogen bonding to other nucleic acids was when we have the codon and the anticodon actually matching up with each other. So it does happen quite often. It is one thing that gives RNA a lot of its unique properties. And also, if we keep in mind that RNA can assume specific three-dimensional shapes, shape is important for function, and the more shapes we can take on, the more functions that we can have inside the cell. There are so many different roles for RNA inside the cell, and many of those different RNA molecules are going to act as catalyst. So as catalyst, we're saying that they do act like enzymes, and specifically, we call these ribozymes. So while the majority of enzymes that we have inside the cell happen to be made of proteins, there are some very specific, very important ones that are made of RNA, and we do refer to them as the ribozymes. This table right here is just a table that's taken out of your textbook. We have talked about almost all of the RNA molecules that you see listed on here. If we just run through them quickly, the messenger RNA was what we actually were sending out to the ribosome. That had the information for making the polypeptide. The transfer RNAs were involved in bringing the correct amino acids to the ribosome. The ribosomal RNA was a component of the ribosome. Now, that ribosomal RNA, that is actually the catalytic part of the ribosome. So it is considered a ribozyme. It is the one that is actually catalyzing the formation of those peptide bonds between the amino acids. So we do want to keep that in mind with regard to the ribosome. The primary transcript was the initial piece of RNA that we made from the transcription process. We did talk then about turning that primary transcript into an mRNA molecule. And to do that, we had to add a cap and a tail, and we had to splice it. Now, it's important to realize here that every primary transcript does not get turned into an mRNA molecule. Many of them do, but many of them do not. So if we talk about the tRNAs and the rRNAs, those are RNA molecules that would also be made through the process of transcription, because any RNA that's made in the cell is made by transcription. In those cases, there are genes in the DNA that are, say, tRNA genes. So that means that they have the information for making a tRNA. So if one of those genes is transcribed, we get the primary transcript, and that primary transcript then just needs to be folded up into the correct conformation to make a tRNA. We also talked about the small nuclear RNAs. They were involved in the spliceosome, and they are considered ribozymes. The SRP RNA was part of the signal recognition particle, so that one was helpful in determining where the polypeptides that were being made needed to go inside the cell. We have a few other types that are listed here: SNO RNAs, small interfering RNAs, microRNAs. We didn't talk about those, but hopefully this table is showing you that we have a lot of different RNA types. And these different RNA types have different roles inside the cell. This is not an exhaustive list of RNA types. So RNA is a molecule that gets quite a lot of research attention. It is very unique, has a lot more kind of interesting aspects, in many people's opinions, to DNA. DNA has one shape to it. It's always a double helix, and it does one thing. It sits in the nucleus and it holds the information for running the cell. But it is a stable molecule that is really just the information. RNA molecules and then the proteins as well are the ones that are really running around and making the cell function the way that it does. We do also want to emphasize the differences between transcription and translation in a prokaryotic cell versus these processes in a eukaryotic cell. If we look at the prokaryotic first, prokaryotic cells do not have a nucleus. So this means that all of those molecules that are inside the cell are all right next to each other. So our DNA is going to be transcribed in the process of transcription. So we will make an RNA molecule, and that RNA molecule will get translated as well. The difference here is that the ribosome is right there next to the RNA molecule as we're making the RNA molecule. So this means that there's no spatial separation, no different compartments that the transcription and translation is taking place in, and that means that a lot of times the time frames are overlapping. So many times while we are still making our RNA molecule, the 5' end of it will be the first end that we synthesize. So once that end is made, a ribosome may assemble on it and begin the translation process right away. That eliminates the opportunity for any kind of RNA processing. So no cap and tail, no splicing is going to take place in a prokaryotic cell. In the eukaryotic cell, which is what we focused on, we do have the nucleus. That nucleus is going to spatially separate the process of transcription from the process of translation. So we make our RNA molecule in the nucleus. We are going to process it by adding a cap and a tail and then splicing it. So essentially we are going to get that RNA looking exactly like we want it to look before we send it out to the ribosome. So the ribosome does not even get a chance to translate the RNA until we are really ready for the ribosome to get a chance to translate it. We did talk about the benefits of the splicing process. We're going to be able to make a larger variety of proteins because of the alternative splicing, and you don't have that opportunity in a prokaryotic cell. The link for the animation that you see listed here, that one is posted as one of your animations on Angel. So be sure to take a look at that particular one. It will show you the processes in motion and hopefully emphasize those differences to you. Now the last thing we want to talk about in this chapter is what mutations are. Mutations are changes in the genetic material of a cell. So the genetic material is DNA, so we're really talking about changes in the DNA. The problem with changes in the DNA is not so much that the DNA now doesn't function properly, but the molecule that we want to make from the DNA may not function properly. So if we go back to our sickle cell example, wild type would be our normal type of DNA, basically our non-mutated DNA, and then we have the mutant DNA. Notice that there's only one letter difference between the two. The difference ends up being a difference in the mRNA after we go through the transcription process, because when we make our RNA, we're working directly off of the DNA template. So if you change the DNA template, you're going to change the RNA molecule that you make, and now that may potentially change the codons, which are going to change the polypeptide that you make. So notice that in someone that does not have sickle cell, we've got this amino acid here, which is glutamate. And then in someone that does have sickle cell, they have a different amino acid there. They have valine. That one amino acid in this case is enough to alter the protein shape, enough that we ultimately end up with a different shape to our red blood cells. So that is an example of a mutation. It is an example of a point mutation. So a point mutation is a change in just one base pair of a gene. So we change just one letter there, but it did ultimately change the protein enough so that it functions a little bit differently inside the cell. Now there's different categories of point mutations. One of them is what we call a base pair substitution. In this case, we're taking a base that was there and we're basically putting something else there. So we're substituting one for the other. There can be different effects of base pair substitutions. If we look at this slide here, the top we have our wild type non-mutated situation. If we change this particular nucleotide right here, so we're putting an A instead of the G that was there previously, notice that that does change the mRNA, and if you look up the new codon, which is now GGU, it codes for glycine. Well, actually that happens to be the same thing that this codon coded for. So in this case, the polypeptide that we make is the exact same. There's no difference, which means that this is going to have absolutely no effect on the individual. So this is what we call a silent mutation. Silent mutations can occur because we have some redundancy in that genetic code. So keep in mind that in some cases, there's as many as 6 codons that may code for the exact same amino acid. As long as the end product, which is the polypeptide, ends up exactly the same, then there is absolutely no harm done. Now that can be one thing that happens. It is possible that it is more dramatic than that. So as we see here, if we change a different nucleotide, in this case this one is changed, now we've changed our codon here, and when you look up AGC, that codes for serine. So now we have a different amino acid there. That's what we call a missense mutation. A missense mutation changes the amino acid that is in the protein. That may or may not cause a problem for the individual. So if we think back to our sickle cell example, that was a base pair substitution that caused missense. It changed one amino acid, which happened to be a critical amino acid for shape. And we did then end up with a protein that was altered in its ability to function. So missense mutations may be bad. They may not make that much of a difference. We have to keep in mind that in some cases our polypeptides may be 2,000 amino acids long, and each one of those amino acids is not going to have an extremely important role in holding together the overall shape of the protein. Now another possibility is that if we change a nucleotide, now when we look at the codon, we actually get a stop codon. This is what we call a nonsense mutation. Nonsense mutations are going to cause the polypeptide to be a lot shorter than what it should have been, because as soon as the ribosome encounters a stop codon, it is going to stop translation. So that means that we will be missing some portion of the polypeptide, and depending on how much of it we're missing, we may very well have a protein that's not going to be functional inside the cell. Those are our base pair substitutions. Here we see just an overview of the missense mutation and the nonsense mutation. Notice with the missense mutation, we're changing one amino acid. The rest of the protein is going to be exactly like it should be. That may or may not be okay. So in the case of sickle cell, we saw that even one amino acid can be problematic when it's changed. The nonsense mutation, as soon as we encounter the stop codon, we are going to stop translation. So we will be missing probably a number of amino acids that should have been there in that particular polypeptide. Now another possibility besides base pair substitutions is that we can get base pair insertions or deletions. So we're talking about inserting extra nucleotides or deleting nucleotides. The difference here is that if we add one or we delete one, we are going to change the reading frame. So in this case, it may shift everything over. In this example, we see an extra A that gets incorporated. Notice that it does shift everything over in the mRNA, and in this case, we actually get a stop codon immediately. So we've shifted our reading frame over, so we call that a frameshift mutation, and that frame shift causes nonsense. If we look at this example, this is a deletion, and when we delete 1 or 2 for that matter, we're going to shift the reading frame. And if you shift the reading frame, now you're not just changing one codon, but you're changing all of the codons that come after that. So this would be an example of what we call a deletion that causes frame shift, that causes extensive missense, because it's the missense of many amino acids. That is probably going to be quite bad for a particular cell, because now, although the protein may be quite lengthy, maybe the majority of the protein is not anything like the sequence of amino acids that it should have been. So it's probably not going to function properly. Now, it is possible to get deletions or insertions in multiples of 3. If you get that, in general, it's not going to be as tragic, because now you're not going to actually shift the reading frame. So we may have an example where we're missing one codon, which means we're missing one amino acid, but the rest of the protein is still going to be exactly the same. So understand that insertions and deletions can cause frame shifts if we're not talking about multiples of 3. If we're talking about a frame shift that occurs in a multiple of 3, then we're just going to be altering 1 or 2 codons and amino acids. Here we see an example where we have a deletion that causes a nonsense mutation. And then on the bottom there, you see a deletion that causes basically a bunch of missense. So now we're changing lots of amino acids that should have been there in the polypeptide that we're making. Know what a mutation is. Understand that the mutation is happening to the DNA, but the problem of the mutation comes in the molecules that are made from the DNA. So getting non-functioning molecules made from the template because the template has been altered. We also want to know the difference between nonsense, missense, and silent mutations, and also the frameshift mutations. Now this slide right here just summarizes really what we talked about in chapter 17 here. The DNA is in the nucleus. We've really focused on the DNA in the nucleus in this case. And that DNA contains regions, so specific nucleotide sequences that we would consider genes. The gene is a region of the DNA whose final product is going to be either a polypeptide or an RNA molecule. So if we need to make a protein or an RNA, there's going to be somewhere in the DNA that has the template for that. When we decide to use that template, we are going to have to go through the process of transcription. So our RNA polymerase will be involved, and that will take place in the nucleus. If we're making a polypeptide, we're then going to take that RNA molecule, we're going to add a cap and a tail, and we're going to splice it. And then we are going to export that mRNA to the cytoplasm. Once it gets to the cytoplasm, it is going to be bound by a free ribosome. That RNA will be translated into a polypeptide, and that polypeptide then will have a number of things that happen to it. It's going to get some post-translational modifications, very likely. It's going to get folded up, and it also has to be shipped to the correct location. If all of those things happen correctly in the cell, then now we've got a functional molecule. All of these processes, the transcription and the translation, these are things that are taking place every moment of every day in every single one of our cells, because every cell needs different things made at different moments in time, and so they're going to be constantly transcribing different genes and constantly translating those genes as well.